Thank you, Linda. And in the interest of time, we conclude here the session. I'll see you tomorrow. Right? Okay, so we've got, I think, 3 speakers in this session, and the second one is online. And so we start with Dr. Bill Snelling from the Food Standard Agency.
Hello everyone, I'm Bill. I'm in the Science and Surveillance team within the Food Standards Agency for Northern Ireland, and thanks very much to Linda and Prachi for the opportunity to speak today. This is a wee bit different from the other talks, so it's kind of AMR surveillance and what I do and my team does for that within the FSA. A wee bit of kind of an overview of what the FSA does, in particular my role, and then my kind of varied career journey to where I am now. So firstly, many people here are probably already aware, the FSA work across England, Wales and Northern Ireland, but just to emphasise as well, we do work closely as well with Food Standards Scotland and then also the Food Safety Authority of Ireland as well too. Our main objective is to protect public health from risks arising from food consumption and to generally protect the interests of consumers in relation to food. Over the past 20 years, for example, with EU exit, developments have resulted in a remit varying across the UK, but one goal remains constant to try to ensure that the UK has food we can trust. So this is a kind of oversimplification of my roles within the FSA. You can see there are AMR and retail meats, but then also a big part of what we do is sample mapping to try and ensure that and avoid duplication of sampling both within FSA in Northern Ireland, FSA on a wider scale across the UK, but then also between government departments as well, because obviously sampling and lab testing is relatively expensive. I'm involved with other projects that don't involve microbiology or AMR, for example, the Retail Surveillance Survey. I lead on that and the deliverance of that for Northern Ireland. That involves about 1,000 retail samples collected across England, Wales and Northern Ireland, 100 of which come from Northern Ireland out of the roughly 1,000 or so. And that involves testing for undeclared allergens, etc., in retail foods. Again, there's some varied rules there, but the main thing is, again, kind of common thread, or one of the common threads, is that we're trying to ensure that food is safe, is what it says it is, and is healthier and more sustainable moving forward. So again, everyone here is probably familiar with this, but why is AMR important? Don't need to say it to this audience, but rising health costs, $1 trillion predicted by 2050. It's a global challenge for all countries at all income levels globally. In the future, C-sections and hip replacements could become more and more challenging if things don't change. But then also for low resource countries, they can probably become more impacted, and maybe already are, by the drivers and consequences of AMR because of poor access to clean water, sanitation and hygiene. So this is something that I've become much more familiar with since I joined the FSA in 2021. That is the UK and NI AMR policy, and in particular, the 5-year national UK action plan, which we're in the middle of, and then also monitoring AMR and retail meats, which fits in with the Northern Ireland AMR action plan. This is just an infographic of that. So my role for AMR and retail meats would be 3 in this infographic, a summary of the national action plan, but then also lots of innovation and influence here as well. So we all have a part to play in this One Health and AMR National Action Plan. So a wee bit more about Northern Ireland AMR policy. DAERA are the competent authority for AMR in animals and are responsible for the programme of abattoir sampling of animals and sampling fresh meat at BCPs. And the FSA, we are responsible for sampling fresh retail meats in Northern Ireland. And specifically that is for ESBL, AMP-C and CP enzyme producing E. coli under mandatory EU regulations. And we contract AFB as well to do our lab testing of these AMR E. coli. Many of them are sitting there in the second row, the audience today, for retail meats. Also, responsible for AMR reporting. So there are monthly updates at harmonised monitoring cross-government meetings where I provide updates on AMR and retail meats and the sampling and testing of those. There's also a research alert system for across the UK for bacterial isolates of major human and animal health concern. And thankfully, there have been none from AMR in retail meats. So, why monitor AMR in retail meats? AMR surveillance is crucial to monitor and understand if retail meats pose an AMR risk to public health and animal health. It's important to continue monitoring over time, to analyse trends over time, and we basically need to, because it's there in the regulations, it's mandatory. So it's part of EU regulation 2201729. That first piece of legislation there dictates, if you like, what needs to be tested, and so AMR and retail meats, and for AMR E. coli. And the second piece of regulation there is basically mainly says that we need to do and complete our EU data returns before the end of May each year. So again, most people there probably know this, but why monitor E. coli? It's a normal commensal, if you like, inhabitant in mammals. Most isolates do not cause observable clinical disease. However, commensal bacteria can be AMR gene reservoirs because of, for example, plasmid gene transfer. And E. coli are important AMR indicators. So, from monitoring AMR E. coli, you know the presence of AMR typically circulating in food-producing animals. This is just a summary of the actual process of our EU data return. So the first part here, the sampling, that's carried out by Hallmark. And basically, our sampling design needs to incorporate population data. So it's according to population density, and from all of the 11 Northern Ireland District Council regions, also determined by market share data, and all of the protocols in this diagram, from left to right, follow EU guidelines and EFSA protocols. So 600 samples are collected per calendar year. We're currently in a year of retail pork and beef. So 1 year it's pork and beef, the next year it flips over to retail chicken and turkey. The meat samples that you analyse have to be fresh, not frozen. They can't be cooked. They can be sliced or diced. They can have skin on or off, but you're not allowed sausages or burgers or kind of processed meats, and you don't have meats marinated in dry spicy rubs or oily rubs or things with kind of chives and flavourings like that. So it's fresh, not frozen. Retail meats, you can have whole turkeys or crowns or legs, for example. So then Hallmark complete the first step there, and then AFB complete the isolation and detection of AMR E. coli on selective media. We then have MIC testing according to EU Cas-ECOF values and whole genome sequencing as well. And the sequences of our isolates of AMR E. coli are accessible from the European Nucleotide Database site as well. We then, well, AFB then perform the data analysis and EU and FSA reporting. Again, the EU reporting is due before the end of May each year. And then AFB write a public-facing kind of AMR corresponding report of that, of the main findings. So our results from 2023, basically low levels of AMR and consumer risk from UK retail meats have been found since 2015 by the FSA when these studies first started. And basically, we found that in 2023. AMR in Northern Ireland retail meats compares favourably with the levels observed in many EU countries based on harmonised surveillance data. It's important that basically the protocols and methods are harmonised so that you're comparing like with like and not comparing apples with oranges, if you like, between EU countries and member states. And low levels of AMR, ESBL, E. coli, 3%, were found from fresh pork and beef retail samples and available for retail sale in Northern Ireland in 2023. The numbers here were not the full required, 300 of each type, because we were working towards, I joined the team recently, and we were working towards that full EU return, which we were able to do the next year. But no isolates sampled were resistant, and this is important, to last-line antibiotics, including cholesterol, and provided good hygiene and cooking practices are followed when handling raw meats, you have a very low risk of AMR-related infections through the handling and consumption of retail contaminated meats. And that's just an example of our FSA report from the 2023 data. And then our full EU return from 2024, that was 600 chicken and turkey samples, 300 of each type. And we had a 12% overall ESBL AMP-C prevalence. And again, AMR in retail meats from Northern Ireland compares favourably with levels observed in many EU countries based on harmonised surveillance data. And again, no isolates sampled were resistant to last-line antibiotics, including colistin and carbapenemase. So again, provided good hygiene and cooking practices are followed, when handling raw meats, you have a very low risk of AMR-related infections through the handling and consumption of retail contaminated meats. And again, just an example of our AMR report with AFB there as co-authors as well. So again, central FSA, so the third research division, do conduct as well research projects on AMR. So this is an AMR in raw salmon fillets that was recently published by our central FSA colleagues, and that involved sampling around 300 samples from across the UK, around 10 of which came from Northern Ireland, and that study found that the risk of Listeria monocytogenes and AMR in UK retail salmon fillets, raw salmon fillets, is low. No salmon fillets had E. coli or Listeria monocytogenes that were resistant to key clinically important antibiotics. And these results provide reassurance for consumers and regulators of the generally low microbial and specifically AMR risk associated with raw salmon on the market. And again, if you follow hygienic cooking and handling practices, you will minimize the risk even further for consumers. So now on to and this is the bit that's a wee bit more awkward, my varied career journey. So I was interested in science at school, didn't really know which one, so I did choose to do a degree in Science with Management Studies at Napier. I quickly realised I liked biological sciences and liked microbiology within that. I then was lucky enough to win a scholarship. I think it was called definitely involved the Department of Agriculture anyway, I forget the exact name, but it was from Northern Ireland anyway, and won a scholarship to do a master's in food science and microbiology at Strathclyde University. And then did a PhD in Campylobacter, and that was linked with the food industry, in particular, Moy Park. So one of the things I learned along the way is to be flexible and adaptable, because my PhD was to look at loads of farms and the Campylobacter population within water supplies. And basically then the foot and mouth crisis happened 5 farms in, and it had to flip to being pretty much completely in vitro for understandable reasons. The other thing I learned along the way was to basically network and join clubs and societies. So one of the great things was Scottish and British intervarsities in Napier and Strathclyde, and then also the Scottish intervarsities in Coleraine, which was a real highlight. And then keeping in touch and staying in touch with friends who made along the way is key as well. Having a 30-year reunion in Edinburgh in January with friends met from sport and from Napier and also Strathclyde. Along the way, who are managers in Northern Ireland Water, Scottish Water, and Merck as well, so public and private sector companies. So make those friends and contacts and keep them going, basically. And one of the good things as well about sport is when you do have stress and things don't go according to plan, it is a good kind of stress buster too, keeps things in perspective. So I then did my first postdoc with Professor Paul O'Toole in University College Cork. That was with Helicobacter pylori, looking at if HORB was an adhesion, lots of tissue culture, generating knockouts. So first did some molecular biology and PCR before that in a PhD, but then quite molecular biology kind of driven and involved. The good thing about that postdoc was it involved collaboration with Limerick University and Galway University, and the Irish division of the Microbiology Society was really good and active, and lots of poster presentations in Trinity and University College Cork and Galway too. The other thing, my next postdoc was with Professor James Dooley in University of Ulster in Cryptosporidium, and a bit of Campylobacter research as well. And the great thing about that was the opportunity to be PI in my first grants. So they were relatively small, I think £5,000 to £10,000 from Invest in I RTD networking grants. But the great thing about those were they opened up opportunities to collaborate with Health Canada for a month, learning more about microarray with Dr. Kathy Sorello and Professor John Nash. You would now use RNA-seq, but anyway. And then also a 3-month research visit as well to the USDA with Professor Norman Stern, who was one of the leading Campylobacter epidemiology scientists there at the time as well. So again, those research grants didn't just happen, had to write them and use the impact and leverage from my publications from my PhD as well, an AAM publication, for example, to get those research grants and collaboration opportunities. And also make the most of it when you're younger as well, because that's obviously much harder if you're married with 2 young kids and stuff to go to America for 3 months. Then as well, spent 7 years in Randox as a senior scientist there. That was using immunoassays to detect drugs of abuse, mainly from human and urine samples. So we would detect the parent drug mainly in blood, and then obviously metabolites in urine. And so that was probably as well my first experience in an audit-rich environment, ISO 13485 for medical devices, for example, and then US FDA audits as well. And again, that was a good experience because that was the drugs that we detected moved from kind of Zaloplan and Zolpidem sleeping drugs to you could really see the designer drugs and spice detection. So that was kind of taken off then too. And again, great opportunities to for poster presentations, etc., and to present at toxicology conferences, for example, in Florence. I was a research scientist for a while at Queen's with oral Musnier. That was looking at rhinovirus. So again, that was my first experience of growing viruses. I think it was with HeLa cells, but anyway. And then data manager as well with mainly prostate cancer trials with the NHS for a while too. That was very rewarding because it was, you could obviously impactful research and improving patient outcomes. So for example, gold fiducials in the prostate area. So during radiotherapy, the dose of radiotherapy was concentrated and less treatment appointments were needed because the gold fiducials absorbed the radiotherapy. And then my final position before where I am now was back with Ulster University as a senior postdoctoral scientist, and that was good again. That was with the Safe Water project, and that project was great because I got to work with Cryptosporidium, E. coli, and then also MS2 bacteriophage, and I helped to and led the water treatment design and trials of water treatment devices to basically check that they zap bugs and kill them. MS2 was chosen because it's very resistant to UV light, and then Cryptosporidium parvum is important because it's very chlorine resistant. So it kind of brought together a lot of the bugs I'd worked with in the past. But then that was also very rewarding because that was working with neglected indigenous rural communities in Mexico and Colombia. And you could see the impact of that with these communities where their children under 5, when they got improved, treated and clean water, their health parameters included, like the Bristol stool chart, and then also those kind of things as well. So their health did improve, and that was very, very rewarding. I think that's nearly, yeah. So why work at the FSA and the science part of it too? So I think the FSA is a great place to work. I may be biased, but we're very supportive, and we use science that's evidence-based, so it's used to inform policy. Many jobs require a science degree, so for example, microbiology or biological sciences. Especially a lot of the jobs are in the central CERD research division. Examples of jobs would be as a microbiology risk assessor, a food safety science officer or lead like myself, and I would recommend keeping an eye out for future jobs on the FSA website. Just want to thank the organisers again for letting me speak today, and AFBI for their great work in AMR and retail meets, and also Hallmark too. And you can follow the FSA as well on social media there as well. So that is me.